Hello teachers, today my group consisting of Joanna, Wani, Jikin and myself, Gordon, will be presenting to you on our model that we've chosen, which is the DNA. After this presentation, we hope you would have learned about the following. Firstly, polymers. Secondly, linkage group between monomers. Thirdly, types of molecules. Fourthly, inter versus intramolecular bonds. And lastly, conformation of a molecule in relation to its functions.
This is our model. Our model is made of statistics. We know some groups choose paper to make the model, but we think paper is not fixable as we cannot twist paper and hold it in one position. Therefore, we choose statistics. And in our model, yellow statistics represent the A and B. Then blue represents the T, timing. Red represents graining, and yellow represents cytosine. Second, our model is made in ratio. As we know, one complete ton of DNA structure is 3.4 nanometer. We use one statistics, which is about 30 centimeter to represent one complete ton. Therefore, in ratio, the diameter of the DNA structure should be 17.6 centimeter. In our model, nucleotide is 7.5 centimeter, and the hydrogen bond in between is 2.5 centimeter. Therefore, in total, the diameter is about 17.5 centimeter. It is quite accurate. Third, DNA structure is a double helix structure. The two sides of the DNA structure represents the sugar phosphate backbones. These are represented by the brown statistics. There are also hydrogen bonds in between. As we know, adenine and thymine has two hydrogen bonds in between. Therefore, we use two copper wires. And there are three hydrogen bonds between the guanine and the cytosine. Therefore, we use three copper wires. There are also strong covalent bonds, which are phosphodiester bonds to link each unit of strand. Therefore, we use a clear tape to hold it together. The structure is in anti-parallel, so one end of the DNA structure should be 5 and the other end should be 3 end. Therefore, on the other side, this side should be 3 end to 5 end. As we know, DNA structure is a stable structure because of three kinds of bonds. The strongest one, phosphodiester bond, the medium one, hydrogen bond, and the hydrophobic interaction in between the nitrogenous bases. But we didn't show in our model. Yeah.
Now I will talk about the basic structure of nucleotides. Each nucleotide is made out of three components: the pentose sugar, the inorganic phosphate, and the nitrogenous base group. The pentose sugar involved in the formation of DNA is deoxyribose sugar. Unlike the ribose sugar involved in the formation of RNA, a hydrogen atom is attached to the 2' carbon of the deoxyribose sugar instead of the OH group attached to the ribose sugar. The second compartment is the nitrogenous base group compartment. They are organic rings that are hydrophobic in nature. They are divided into two groups, purine and pyrimidine. Purine consists of a 6-side carbon as well as a 5-side carbon ring. Examples of these are adenine and guanine. Pyrimidine consists of only a 6-sided carbon ring. Examples of these are thymine, cytosine, and uracil. However, uracil is only involved in the formation of RNA, while thymine is only involved in DNA. Purine and pyrimidine are able to form hydrogen bonds with one another in a very specific manner. The third compartment of a nucleotide is the inorganic phosphate. It is derived from the phosphate acid PO4 2- and give rise to the negative charges on the nucleotide.
The three components form chemical bonds with one another at specific sites by means of condensation to form nucleotides. There are two condensation reactions involved. First, the nitrogenous base is joined to the 1' carbon atom of the pentose sugar with the elimination of water.
The product of this condensation reaction is called a nucleoside. Then, further condensation will occur between the nucleoside and the phosphoric acid.
The phosphoric acid is joined to the 5' carbon atom of the pentose sugar with the formation of a phosphoester lineage. In total, 2 molecules of water are removed in the formation of the nucleotide.
structure of a DNA strand, the phosphate of one nucleotide is attached to the sugar of the next, resulting in a backbone of alternating phosphates and sugars from which the base projects. The polynucleotide strand is directionally from the 5' end with the phosphate group to the 3' end with the OH group. 5' and 3' end referring to the numbers assigned to the carbons in the sugar ring. The diagram on the board represents a flattened out model of the DNA strand. If you notice the legend on the board, there are a few bonds present in the strand. Firstly, there's the phosphodiester bond, which is green in color, which serves to link nucleotides together. It is also known as the backbone of the DNA. The phosphodiester bonds are made out of very strong covalent bonds. Secondly, there are the hydrogen bonds, red in color, between adenine and thymine, and guanine and cytosine, which are formed in a very specific manner. Take note that A and T have 2 hydrogen bonds, and G and C has 3 hydrogen bonds. The ratio of A to T and G to C is always 1:1, and the 2 chains are complementary. Thirdly, there are weak hydrophobic interactions, which are black in color, between the nitrogenous bases. Lastly, there are the phosphoester bonds, which are dark blue in color, which links the inorganic phosphate group and the nucleoside together. Just to recap, the nucleoside is made out of a pentose sugar and a nitrogenous base. Do not confuse the phosphoester and phosphodiester bonds as they serve separate functions. Thank you, teachers, for viewing our presentation. We hope you have learned more about the aforementioned objectives as stated in the introduction. Thank you.